Hi guys, this is the first of my series of little videos walking you through your independent lab assignment. So again, this is replacing the week of lab where I'm out of town and kind of getting you guys ready for your PCR-based project that you're going to do for the second half of lab. So the goal of that project is to use PCR to determine if your genomic DNA samples are contaminated. And by that, we mean if they have anything other than E. coli in them, the E. coli we started with. So first things first, as we work our way through, is remembering what PCR is and how it works. So these are the slides from class. It'll be last week when you start this. And so there's two main things that you really want to remember about PCR when you're thinking about this project. And the first one is what region of the DNA is being copied. So I have this figure up here. So here's our original strand of DNA. The cycling process that happens during PCR is denature, which means essentially melt the DNA fragments apart from each other. So these two light blue strands get separated, one up here, one down here. The second step is annealing, which is where the primer is going to base pair. And so the primers in this figure are these little reddish orange pieces. And then the third step is extension, right? Where you build DNA off of that primer. And so what's really important to remember is that what we're going to copy is the region between the two primers. So it's hard to conceptualize a lot of the time because they never draw the DNA beyond the primers. But remember that the genome of E. coli is 4.5 million continuous base pairs in one big circle. And so they're only being lazy, right, and showing you this section that's getting copied. But effectively, here's one primer binding to this strand, the other primer binding to the opposite strand, and we fill this in, right? And eventually we end up with all these pieces of DNA where we have copied and made many, many, many copies, right, of the DNA in between the two primers. So that's the first really important thing to remember because you're going to start thinking about, okay, what are my primer sequences? What section of the DNA am I going to copy? And how long is that piece of DNA actually going to be? So remember that. The other thing you really want to remember, I'm going to skip ahead here. They show the primers a little bit better here with a little extra DNA sequence. And so the region to be copied, again, is between the two primers, as you can see in this image, right? We're not going to end up copying any of the bases beyond the primers on the outside, just between. That's point number one. Point number two is the primers have to match. Okay? So here they're showing nice, beautiful, perfect primers where they have been designed to identically match your DNA sequence, and every single base matches. If this sequence is different in a different bacteria, the primer won't match and it won't bind and you will not get a PCR product. So those are scenarios that you're going to be looking for later on when you look at what are the DNA sequences in different bacteria, what are my primer sequences, how is that allowing me to differentiate between different bacteria? And it's because the primer sequence has to match to be able to bind to get a product. So if you have a different sequence in a different bacteria, that's not going to happen. All right, so keep those two things in mind as we move forward.